Hi guys, in this video, we are going to do the preparation for your UAS, paper 3, Ujian Amali Sains, paper 3 for biology paper 3. So the sample topic that I've chosen here is the effect of carbon dioxide concentration on the rate of photosynthesis of hydrilla plants. Okay, so again, I want to stress, this is not a prediction, it's not Ramalan. Please do prepare how to answer if this topic doesn't come out. Don't panic if it doesn't come out. It's all right. Because from the techniques that you will learn, you can answer, even though it is not the topic that has come out. Right? So let's look at this. State the list. So first of all, the first thing that we need to determine every time you get your question in your paper 3 is your MV and your RV, manipulated variable and responding variable. So you are required to carry out an experiment to determine so when you get this type of question, then it becomes a bit simpler. So to determine what? The effect of. So this is one type of question, the effect of. So effect of, usually after effect of will be the MV, carbon dioxide concentration. And on what? The rate of photosynthesis will be RV. So usually this format of question is to determine the effect of MV on RV. Okay, so this is how you determine your MV and RV. Again, as usual, which is MV, which is RV? MV will be what we change at the beginning of the experiment. RV will be what we collect at the end of the experiment. Okay. Now, first one, state the variables in this experiment and the method of handling the variables in the table below. So you will notice from the many trial papers that are available for this year that for bio specifically, there's lots of questions on method of handling variable. All right. So in this video, I've prepared some sample answers for you guys to follow. Unfortunately, we don't really have mark schemes that have the complete method of handling variables, all the variables. So this is my sample answer. All right. So for the variables, there are no doubt for the variables because this experiment is in your textbook. So here, manipulated variable is the concentration of sodium hydrogen carbonate solution, not carbon dioxide concentration. Carbon dioxide concentration is the MV in theory, but practically, how do we manipulate the carbon dioxide concentration? Practically, what determines the carbon dioxide concentration? It is actually the sodium hydrogen carbonate solution. So again, we can be confident in this. I can confidently tell you this because this is how it is in your textbook. Okay, they don't use carbon dioxide concentration. Okay, so let's go on to the method of handling this variable. So method of handling the variable is very simple. It doesn't require you to write a full procedure. It's usually just 1 or 2 lines. Okay, so specifically how do you manipulate the concentration of the sodium hydrogen carbonate solution? How do you manipulate it? How do you differentiate it? So the answer here would be to use different concentrations of sodium hydrogen carbon solution. That's all. However, do state the specific values. So for example, 0.02, 0.04, 6, 8, and like this. Do state the specific concentrations that you're using. Now, in the textbook, we have it in molar. This is mole per dm cube. Okay, how many molar? They also could give you in terms of percentage. All right. So please read the question carefully and apply accordingly. Don't copy this exactly. You must adapt to the question. Okay. Nobody knows what the exact question is going to be like. Nobody knows if this is actually going to come out. Right. But even if it does, please do read the question properly and follow what is in the question. Okay. What is the responding variable? Responding variable was rate of photosynthesis. But once again, this is only in theory. Practically, how do we determine the rate of photosynthesis? So this is the reason we are using an aquatic plant, which is hydrilla. Hydrilla is an aquatic plant. It lives in water. So when photosynthesis is taking place, it uses up the oxygen that's dissolved in the water and produces carbon dioxide. Now, this carbon dioxide is going to be, it uses up the carbon dioxide in the water for photosynthesis and then produces oxygen. So this oxygen that is released we can see the oxygen being released as colorless gas bubbles. Okay? We can actually see it. So we are going to assume that the rate of the release of the gas bubbles is equals to the rate of photosynthesis, or rather it is proportional to the rate of photosynthesis. The rate of gas bubbles release is directly proportional to the rate of photosynthesis. So this is what we are going to use to measure the rate of photosynthesis in this experiment practically. So when you give your variable here, give the practical variable. That would be the number of air bubbles released in 5 minutes. So according to the procedure in the textbook, we set the time. We set the time to 5 minutes every time. And then you count the number of air bubbles that are produced within those 5 minutes. Okay. So there's no need to calculate rate after that. There's no need to calculate number of air bubbles over per unit time. No need. It's just the number of air bubbles. Okay. So how do you handle this variable? How do you handle the number of air bubbles released in 5 minutes? This responding variable. So responding variable, the method of handling is always either you observe or you measure. This is usually the responding variable method of handling. Either you're going to observe it or you're going to record it. So in this case, we have the number of air bubbles. So this one is a little bit unique. You're not going to measure the number of air bubbles. You are also not technically going to observe it. You're going to count it, right? We're going to count the number of air bubbles released in 5 minutes. So that's exactly what we're going to write here. So with RV, make sure you add and record. Count and record number of air bubbles in 5 minutes. Okay, so that's how we handle the responding variable. Now, what about the fixed variable? So what needs to be fixed here? This is an experiment on the rate of photosynthesis. So what must be fixed? There's all the other factors that can affect the rate of photosynthesis. So in this case, there are a few answers, but this one I've chosen the type and size of hydrilla species. Okay, type and size of hydrilla species. This is one of the variables. You don't have to mention all here because if you need to mention method of handling the variable, just pick one will do. So how do we fix the type and size of hydrilla plant? How? That is the method of handling. How to fix it? Very simple. We simply use the same type and size of hydrilla species for all the different concentrations of sodium hydrogen carbonate solution. That's it. That's how you fix it, isn't it? Right? So this is how we do method of handling variable. Very simple. Right? There's no need to write lines and lines of procedure. That's not how you do it. Okay. So thankfully, there are some mark schemes existing for this year that have samples of method of handling variable. Yeah, so we can refer to that. That can be a good source of reference on how you should answer this. Okay. All right. Now, next, state the hypothesis. So once we have our MV and our RV, we can state the hypothesis. However, hypothesis is supposed to be a guess. Okay? Hypothesis is supposed to be a guess. So this one, in the textbook, they don't use the practical variables for hypothesis. They use the theoretical variables. Okay. So when we are stating the hypothesis, we go back to the question here. Carbon dioxide concentration and rate of photosynthesis. Okay. Once again, this is purely based on what's in your textbook. And that would be the best reference to follow. Okay. That's all I can say. All right. So for hypothesis, we can use the usual format. When MV increases, RV increases, right? So here again, we use the theoretical variables. So when the concentration of carbon dioxide increases rather than the concentration of sodium hydrogen carbonate solution, when the concentration of carbon dioxide increases, the rate of photosynthesis increases, right? Okay, so tabulate the data. What is the data that we are collecting? So as usual, when it comes to the table of results, the standard table, the simplest of tables, should have manipulated variable in the first column and then the responding variable in the second column. This is how it should be. However, if there is any other data that we are collecting throughout the experiment, it must be tabulated as well. Right? So in this case, what data do we have? We are only counting number of air bubbles. We are not collecting anything else. So there's no problem here. There's only two things you need to record. However, depending on your procedure that you get, you have to see if the procedure requires you to repeat. So in your textbook, we do repeat. We do repeat twice, which means we have a total of 3 readings. And therefore, when you have a total of 3 readings, you need to calculate the average. That's exactly what's going to happen here. So manipulated variable will be concentration of sodium hydrogen carbonate solution. Sodium hydrogen carbonate solution. And the RV on the right will be number of air bubbles released. But since we are taking 3 readings, we need to put it like this. Number of air bubbles released in 5 minutes. Reading 1, reading 2, reading 3, and then the average. Okay, concentration of sodium hydrogen carbonate solution. Again, here I've given in molar because this is what it is in the textbook. You may get percentage. Please be careful. So fill it up accordingly. Now, this concentration is a stated concentration. It is not measured. You don't have to worry about the number of decimal places. Just follow whatever is given to you. Okay. Number of air bubbles, again, this is going to be an integer. So no problem with number of decimal places in this experiment, right? Okay, so this is how we tabulate the data. Now, in biology, you will get this type of question. Based on the results, state one observation. Okay, state one observation. Okay, so what are we going to observe in the first place? Now, let me show you the setup for this. Okay, let's go to the theory for a little while. So if you look at this, so this is the effect, the environmental factors that affect the rate of photosynthesis. Okay, so we have graphs here. Okay, this is the experiment. So in the rate of photosynthesis, we have a few experiments, right? First, we have light intensity. So how do you set up the apparatus? So let's just look at this apparatus setup for a minute, right? So we have the thermometer that's dipped inside here. You may or may not have a thermometer. But this is where the hydrilla is. We place a clip at the bottom of the hydrilla plant to keep it grounded, because usually the hydrilla plant, its roots are in the soil at the depth of the body of water, right? But here, that's not the case. So you need to attach some sort of weight to keep it down. So that's why we put the paper clip there. Okay, then we have a boiling tube here because it's larger. Inside here, we have the sodium hydrogen carbonate solution. So here you can see they use 0.2% because here this experiment is on light intensity, right? So they use a fixed concentration of sodium hydrogen carbonate solution, which is 0.2%. Okay. Okay, then we have distilled water here and a beaker. So the distilled water, this is essentially a water bath to keep the temperature constant. Okay, and then we have a beaker. Okay, so this is the setup. Now, the light source will be about, you will probably get this. You will probably get about with batteries. So there's a light bulb that is placed at a fixed distance. So you can see the meter rule here. You have to measure the distance. Okay, so if we are doing an experiment on the effect of light intensity, by the way, class, this is my normal notes in class, my class that I teach. If you want to join my class, you can go to mrrueltuition.com. You can join. I have SPM preparation classes, yeah, going on right up to SPM. I have two classes. I have an exam drills class at night where we focus only on exam questions as well. If you're interested, please do join. It's a very simple process. No need to contact anybody. Just go to the website. You can straight away get everything you need, right? Okay, so let's continue here. So if the experiment on light intensity comes out, then how do we manipulate the light intensity? By placing the bulb at different distances from the hydrilla plant. So that would be, for example, in this experiment, it would be, what is that? Yeah, 30, 40, 50, 60. So I've taught my students how to answer this method of handling variables and precaution step. Okay, you can pause the video and look at it if you'd like. This is my sample answer. Yeah, this is not any official answer. This is my sample answer. We don't have any official answer. Okay, so that's how you would manipulate light intensity here. We won't change the power of the bulb, but we change the distance. However, in the actual SPM exam, they most likely will modify the experiment a little bit. So you may be asked to, for example, use different number of batteries each time. Instead of changing the distance of light source, you may be asked to use different number of batteries. In fact, that's one of the questions that came out for one of the trial papers, right, this year. So yeah, that is possible as well. Okay. So this is one setup. This is for light intensity. Then the setup for the rest is the same, but when it comes to temperature, of course, the only thing that we do is you will have to add a water bath. It's already in a water bath, but we have a Bunsen burner at the bottom so that we can manipulate the temperature. So they get heated up to different temperatures, maybe 30, 40, 50. Okay. All right. Then 10, 20, 30, 40, 50. Okay. And then we also have this carbon dioxide concentration, which is what I am focusing on here in this video. So for carbon dioxide concentration, the setup is the same. And this is how it is in your textbook. So we have 0.01, 0.02, all the way until 0.10. I doubt you would be made to use so many different concentrations. There is no time. Yeah, so each one you have to count for 5 minutes. This is 10 concentrations. That's already 15 minutes, more than the 45 minutes allocated. You will probably do 3 to 5. Okay. Yeah. So that's how we do it. So this is the setup. Basically, this is the setup. This is how it's going to be. Okay. So if you are manipulating the carbon dioxide concentration, we are actually going to change this, the concentration of sodium, hydrogen, carbon solution. Right? So you're going to count the number of gas bubbles that form inside this boiling tube. Okay. Okay. Now back to the question. So observation, the question specifically is state one observation from the experiment. This is very common, okay, in bio. So you look at this data, they just randomly ask you to state an observation. They didn't ask for any specific manipulated variable. So you choose one of the manipulated variables and state. Usually we go for the extreme values, the highest value or the lowest value. Choose the highest or lowest value to state as an observation. So here we can use what I've chosen here is the lowest, okay, 0.01. So this is my sample observation. When the concentration of the sodium hydrogen carbonate solution is, and the concentration of sodium hydrogen carbonate solution is 0.01 molar, the lowest concentration, the average number of gas bubbles released in 5 minutes is, give a specific value. Give a specific value. So this one depends on your own experiment. So whatever average value that you got here, use the average value. Okay. So the average number of gas bubbles released in 5 minutes is whatever value you get from your experiment. Okay. So now we need to state an inference. So always remember, inference is simply, why is the observation like that? Why for 0.1 0.01 molar, we get this many bubbles in 5 minutes. Why? Okay, so the explanation here would be very simple. The number of air bubbles released in 5 minutes is low. Okay, this is because the concentration of carbon dioxide is low, causing the rate of photosynthesis to be low. So in bio, your inference should be a little bit more elaborate compared to your chemistry. Okay, so here the number of air bubbles released in 5 minutes is low. This is because concentration of carbon dioxide is low, causing rate of photosynthesis to be low. Okay, so we can see this type of format of answers in the mark schemes this year. You can see, all right, when you go through them. Okay, so this is my sample answer for observation and inference. Now, draw a graph. So draw a graph of number of air bubbles produced against concentration of sodium hydrogen carbonate solution. Okay. So of course, the axis, y-axis should be the number of, okay, y-axis should be the number of air bubbles produced. There's no unit, of course, number of air bubbles. And concentration of sodium hydrogen carbonate solution. So again, I'm using molar here. Please check what your particular experiment requires. Okay, or what is given to you rather, what is provided. Okay, so what type of graph do we expect here? Is it a straight line? Is it a curve? So this is, don't forget, this is an experiment. Theoretically, we are looking at the rate of photosynthesis, which should be proportional, directly proportional to the number of air bubbles produced, and concentration of sodium hydrogen carbonate solution, which should be directly proportional to concentration of carbon dioxide. So theoretically, I can only say theoretically, okay, because it varies from experiment to experiment. It depends on which portion of concentration that you're using. But theoretically, these are the graphs. So when it is light intensity, you should get a straight line. However, if you're doing distance of the light bulb from the plant, then, okay, let me show you. I have one here. Okay, look at this. Yeah, okay. So if we are doing distance of light source from the hydrilla, then here the graph that you should get is like this. Okay, it should be inversely proportional. Distance of light source. It should be a downward curve. All right. Okay, so this is the type of graph you should get for distance of light source. Now, for temperature, again, we have to refer to which, the reason why I didn't give my students exact graphs here, because it depends on what range you are given during the experiment. Yeah, so we can only look at the theory. Let's go back to the theory. So according to the theory, this is the graph that you should get for carbon dioxide concentration, which is what we expect in our the sample topic that I've chosen. So you should get a curve that's curving upwards. So again, it depends on which range you're given. If you are given this portion here, then you won't get a curve. You would get a straight line, right? So that's why I say it depends. It depends on which portion that you get. Don't panic if you suddenly get a straight line. It's fine. Maybe that's the range that you got. We don't know. We don't know. It's hard to say. But over a large value of a large range of values, you should ideally get a curve. Okay. Now, when it comes to temperature, if it's a difference in temperature, if it's the effect of temperature on rate of photosynthesis, then once again, it depends on which part of the curve that you get, isn't it? Which range of temperature are you using? Right? If you use from 10 to 40, for example, then you will get the entire thing like this. You should get something like this. But if you are doing 10 to 25 or 5 to 25, 5, 10, 15, 20, 25, then you will get a graph that's curving upwards. So it all depends. It all depends. Just remember, optimal temperature is 25 to 30 for plants. It's not like us for animals, for humans, 36.9. No, 25 to 30, generally. Okay? All right. So these are the different graphs that you may be required to draw depending on what question comes out. So again, the actual graph here would depend on which range you get, right? So that's the graph. Now, based on the graph, state the relationship between the concentration of sodium hydrogen carbonate solution and the number of air bubbles produced. So again, based on the graph, it should be either you will get a straight line like this, or you may get a curve curving slightly like that. I doubt you will get the full, because you won't reach the saturation point. Okay, so or rather, sorry, you won't reach the point where the concentration of carbon dioxide is already maximum, for which the other factors start becoming the limiting factors. I doubt you will reach that. So your graph will probably either be a straight line or a slight curve like that. Okay. Now, here, what is the relationship? Either way, when the concentration increases, as it increases, what is happening to the number of air bubbles produced? Increases as well. So that's the relationship that we're going to use. So when the concentration of carbon dioxide increases, okay, the concentration, the rate of photosynthesis increases. So here, this one is an answer from the MV, but we must answer the question because they state the relationship between concentration of sodium hydrogen carbonate solution. So of course, we just follow this. This one I just copied from the MV first. Let me correct that. Number of air bubbles produced. Yeah. Follow the question. Follow the question. Number of air bubbles produced increases. Done. Okay. So this is how you answer that based on the graph. Now, so I've included one possible. Okay. So this is not something that I included. This is in fact one of the discussion questions in your textbook. Why is sodium hydrogen carbonate solution used instead of distilled water? Why don't we just put the hydrilla plant in distilled water? Why do we use sodium hydrogen carbonate solution? That's because the sodium hydrogen carbonate solution is the one that will provide the carbon dioxide to be used by the hydrilla for photosynthesis. That's why we use sodium hydrogen carbonate solution instead of distilled. Okay. So again, this is one of the discussion questions in your textbook, right? So as usual, discussion questions in the textbook, there are no answers provided. So this is my sample answer. Okay. Now, predict the number of air bubbles. So I've included one KBAT question here for you. Predict the number of air bubbles produced if the hydrilla species plant is replaced with another hydrilla species plant, but with a greater number of leaves, more leaves. So here we have to go back to the theory. The organ that carries out photosynthesis is the leaf, right? So if you have more leaves, what do we expect? Of course, we would expect the rate of photosynthesis to be higher, right? And therefore, what is the effect on the number of air bubbles produced? It will increase. That's it. Done. Okay. All right, guys. So this is the end of this discussion here. Again, don't expect this particular experiment to come. It may not be the case. We don't know. Nobody knows what's coming up, right? It may come out, it may not come out. If it doesn't come out, don't panic. Just use the techniques that you have learned in this video. For example, method of handling variables, okay, the format for hypothesis, the how to tabulate. Just remember all the techniques and you will be fine. Don't worry. Okay. Now, once again, if you want to join my classes for SPM preparation, you can just go to mrrueltuition.com. All right. Okay, guys. All the best to you. I really hope that you'll be able to answer everything. All the best.